Previously, we have seen the identification of exon, how we can identify the exon within the genome. Now we will see the exon chaining algorithm, and in this algorithm, S1 represents the length of the longest path in the graph that ends at the vertex Vi. So the S2n is the solution of the exon chaining problem. So here we can see the exon chaining algorithm. It starts with we assign the value of 1 to 2n to I, and if it is this, then we will assign the value of 0 to Si, and again we will use the for loop and assign the value 1 to 2n for the I, and we know that the value of 2n we have already studied previously, that we have two n values. One is corresponding to the right end and the other is corresponding to the left end. So in the fourth step we can see that there is a if condition, and that is the vertex Vi in G correspond to the right end. As we have seen, we have two ends, right end of an interval 1. And similarly we will assign value index of the vertex to the left end, and that is assigned to the j, and w is the weight of the interval. For the Si we will assign the value maximum value, that is Sj plus w plus Si minus 1, and else and this and that, and we will finally get the value S is 2n. So in this algorithm we can see that the solution of this algorithm is that we have assigned the values to the right and left side of the Elmer's, and then at the end we will get the value S2n. One shortcoming of this algorithm is that we do not get the conserved endpoints of the exons, and this algorithm also do not allow for this one. And another problem of this algorithm is that the optimal chain intervals, they do not correspond to any of the valid alignment, and they do not correspond to the location within the proteins. For example, if there is a first chain interval, it may correspond to the suffix location of the protein, and if we consider the second conserved chain interval, it may correspond to the prefix of the protein. So if we combine these exons and will try to get the alignment, we will not get the proper alignment. So here we can see this is the first exon number 1, and this is the first conserved chaining interval, and we can see very easily that it is corresponding to the suffix part of the protein. And this is the second conserved chaining interval, and it corresponds to the prefix part of the protein. So we cannot get the proper alignment if we combine these two with each other, because it will not correspond to the location of the prefix and suffix present within the protein. So this type of problems is obtained when we use this exon chaining algorithm.